Hi, this is Lisa from biologyforbelievers.com. In this video, we're going to be discussing experimental design lesson 1. As this lesson is actually building off of our discovery science lesson that we did the last video. All experiments, all discoveries start with an observation. In this case, we're looking at these group of flowers and that the water has turned a little bit bluish purple. And so we come up with some questions. Our questions could be, why is the water turning colors? What would happen if I put a flower in colored water? From that and from our discovery science, we develop a hypothesis as we're doing actual experimental design. At this level, our hypothesis is going to be in the if blank something happens, then I predict something else to happen. This is a prediction. And we can base our prediction off of our discovery science. We've had a little bit of interaction between flowers and colored water, and so our prediction, our hypothesis would be, if I put a white carnation in blue colored water, then it will turn blue. So now we have our procedural details that we're going to follow and be very specific so others are able to, if they wanted to, copy our experiment. We have chosen a carnation. The color is white. The color of the water is going to be blue. The intensity of the color is going to be about 20 drops, exactly 20 drops of food coloring in 1 cup of water. And we have learned that we want to watch it for 5 days to make sure that we see the full result. All of this knowledge came from our discovery science. And we want the length of our stem to be 10 centimeters. Now we have our control. What would we need to have set up to make sure that the carnation would just not naturally turn blue? We want to make sure that the color of the water is what is actually turning our carnation blue. Otherwise, our experiment isn't working. So our control would be you do the exact same thing except you change the variable, one variable. In our case, we're going to have a white carnation, 10 centimeter stem. We're going to watch it for 5 days. But our control is going to be clear water. We have to still put the flowers in water, otherwise they will wilt and die. And you want to make sure that everything is the same except the color of that water. Now we're going to talk a little bit more about variables and just define them a little bit better. Usually we don't talk about this until about the high school level, but I wanted to add it in this lesson because I think it's really important to understand where we're going and where scientists are going when they have actual experiments. So the experimental variable is what you are changing to look at. So what are we changing in our experiment? In our case, we change the color of the water. We have clear and we have blue. So our experimental variable is the color of the water. Next, you have your dependent variable. Your dependent variable is what you're going to measure. It's what is dependent on the experiment. So what are we looking at that is dependent on the experiment? And that's what you're measuring or looking at. In our case, we're looking at the flower color. So our experimental variable is the water color. Our dependent variable is the flower color because we want to prove that the flower color is dependent on the water color as we're discovering those questions that we were looking at the beginning. So you can go through what sample size. How many flowers do you think you need to do the experiment on? Do you just need 1 or 2 or 1,000? In science, a really good minimum sample size is 30. That is acceptable. But please recognize there are plenty of experiments that are still published with smaller numbers. And again, we want to make sure that we recognize that there is good science out there. There are good experimental designs out there. We just want to be able to recognize what they are. Sample sizes are important. If we were just looking at one flower, what your observations are may not always be true. And so if you have a minimum sample size of 30, that makes it more legitimate for your variables. So you have 30 flowers. What do you do with them? How do you divide them up? You have the experimental ones, which are going to get the blue food coloring, and you have the controls that are going to get the clear water. You have something called randomization. And again, this is probably along the lines of the high school college level experimental design, but I really don't think it's too advanced for even older grade schoolers to understand. Meaning you don't want to have bias in your experiment. Think about it this way. If I had a bunch of white carnations that were already kind of blue and I want my experiment to work, I can't, what you don't want to happen is to put those in blue water and say, oh, look at, they worked. You want to be able to show that just the color of the water is changing the color of the flower. So we do something called randomization. You pick a flower, you have a coin. If you flip it and it's heads, you're going to put that white carnation into blue water. If you flip it and it's tails, you're going to take that carnation and put it into clear water. This takes out your bias. So you as an experimenter aren't biased to trying to make your hypothesis be true. Please recognize scientists are people too, so there are plenty of scientists that have been biased and have tried to prove their hypothesis with their data. And so this is why, as we're designing experiments, we want to make sure that we have randomization, or we're taking out our desire to want our experiment to work. So here we are. We have our 30 carnations. 15 of them are in blue, 15 of them are in clear water. So we've watched them for 5 days, and now you want to review your results. What do you do with this information? Your prediction is that the white flowers in the blue water will turn blue, and also that the white flowers in the clear water will stay clear. Still stay white. If this happens, fantastic! Your hypothesis was true. You have proven your hypothesis true. At this level, we're going to say that. Now, let's say you had some surprises. Let's say all your carnations are still white. You see no blue. They're all still white. What do you do? Did your experiment fail? Kind of. You can look at it that way, or you can look at it as you need to try again. You redesign your experiment. You may want to try a daisy. You may want to try to put more food coloring in, 240 drops to 1 cup of water. So you go back and you try everything again. Yes, you get 30 more carnations. You do the randomization where you make sure that you're not being biased to where you want to put the flowers, and you do your experiment as you are discovering what happens, your question, what happens to flowers if you put them in colored water. This is the end of this video. If you have any more questions or need more information, please check us out at biologyforbelievers.com.